besides research, I just put this there to say, besides research, I also involve myself in the community. It's not just about the research and the science, but then I also try my best to be involved with everyone around me and to be involved with things that, have to do with community. So to the topic of today, and I'll try to make this as brief as possible, so that I can give room for more questions. And, I know that we have other engagements today. So I'll just try to be concise. Speaking on antimicrobial resistance and tolerance in the food chain. And I would like to start by talking about different stressors that microbes undergo. So in the environment, microbes can undergo oxidative stress. There could be nutrient limitations. There could be oxygen deprivation for some microbes. And even the presence of oxygen for some other microbes can, lead to stress to them. There could be, temperature stress. And then there could be antibiotic stress. I mean, we use a lot of antibiotics and a lot of antimicrobial in the food chain to try to eliminate microbes, especially those that could be harmful. So if we look at the field, these antimicrobial genes or antimicrobials that are used, against microbes can result to the formation of antimicrobial genes in the microbes. So basically, microbes can figure out the way to adapt to these antimicrobials, and they could, take up genes that help them, combat or defend themselves against the antimicrobials. And these genes can be passed from one microbe to the other. So, when you get to the environment, cattle is a big reservoir of microbes. So, cattle can shed as much as 8 billion cells of E. coli into the environment. And even other microbes like Cyclospora cayetanensis, they're all housed in the cattle in cattle intestine. And so they can shed this into the environment. It can get to the fields. It can get to things like feeding trough, it can get to water, and really it's a cycle. So, the cattle can feed on the grass and they can take up bacteria with those antimicrobial genes, and, it can when they excrete them into the environment, it can get on top of crops, and, it just keeps going. So when we talk about antimicrobial resistance, it's not really a surprise that it's a big issue today, because we look at the number of or the amount of antimicrobials that are used in the food chain. So one thing we know of, certain is bacteria can develop resistance to antibiotics. And some studies have also shown that when they develop resistance to one class of antimicrobial or one class of antibiotic, they can become resistant to other classes, especially if the mechanism is similar, which I'm going to touch on a little bit. Now, I pulled this from, data in the United States, and I'm very sure similar data exists for Nigeria. But just in the United States, we have over 25 million kilograms of medically important antimicrobials or antibiotics that are used. If we just focus on cattle production, between 2016 to 2019, there are over 10 million kilograms of antibiotic. And, bacteria, as I said, when they're exposed to these antimicrobials, especially at sublethal concentrations, they can develop resistance. So by the right, I just showed a little bit of a chart on how these genes can get to microbes and get to our food. So you have a cattle that takes up bacteria that has these antimicrobial genes. The bacteria transfers the gene to other microbes within the intestine of the cattle shares it into the environment. And when it shares into the environment, it gets on, the grasses, and other cattle can consume it. Also, when the cattle is slaughtered and used for production, then it can be consumed by people. And, that way we take up bacteria with antimicrobial genes and we can shed them back into the environment. Also, cattle feed can get to manure, manure can get to water, water is used to, irrigate fresh produce. Again, it comes to humans. So it's really a very big cycle of transfer of genes from one microbe to the other, from one host to the other, and it just keeps going. So This chart, I put this just to show us the different classes of antimicrobials or antibiotics that are used. So we have some that target the cell wall synthesis. There are some that target protein synthesis, some target nucleic acid synthesis. And some of the names here, we probably know them. So the penicillins are very common. Tetracyclines are very common, streptomycins are very common, So these are all the antibiotics and all the different classes that are used. And again, based on the mode of action or based on the mode of activity of the antibiotics, you can have cross resistance between, one class and the other. And again, the chart to the right just shows us the complexity of this. So antibiotic use into the environment, irrigation water, on produce, on meat, and that way the cycle continues. So really, the research I'm going to present today was a question that, came up, and it was, okay, we know that bacteria can develop resistance to antibiotics, but how well does this resistance impact cross-tolerance to other classes of not just antibiotics now, but antimicrobials? We need to bear in mind that a lot of antimicrobials are used in food production. So, you have things like quaternary ammonium compounds, you have things like lactic acid. They are all used as sanitizers in the food industry. And so it's like, if we have this concept of antibiotic resistance in the food chain, and if we know that one class of antibiotic can result to cross-resistance to another class, can we also have cross-tolerance to other antimicrobials besides just antibiotics? So things like the sanitizers that are used in the food chain. And we looked into the literature and we found several papers that kind of try to articulate this concept, the concept of cross-tolerance and cross-resistance. So for the sake of this, I just outlined certain definitions that would be very helpful. So MIC is the minimum inhibitory concentration of an antibiotic. So basically the minimum concentration required to prevent growth of a bacteria, resistance. So it's the ability of organisms to grow at concentrations above the MIC. An example, if a microbe if the MIC of, let's say, streptomycin is 20 microgram, and this is just an example, if a bacteria is able to grow above 20 micrograms, then you can say the bacteria is resistant to streptomycin. And then tolerance is a different concept, is the ability of bacteria to survive at concentrations that far exceed the MIC, not necessarily grow, but they're able to survive. So an example, if we put, let's say, 500 cells of E. coli into streptomycin, and if we still have 500 cells of E. coli after exposure to concentrations that are way above 20 micrograms, as I gave as an example previously, then we can say if that specific strain of E. coli is tolerant to streptomycin. Not necessarily resistant, but tolerant. And again, I'm happy to answer any questions that come up along the line when I'm done with the presentation regarding this. So persistence is the ability of a bacterial subpopulation to survive antimicrobial exposure. So really the hypothesis for this study was that antibiotic resistance in E. coli will result in certain phenotypic changes in the bacteria and could result to cross-tolerance to lactic acid. And the specific objectives were to determine the influence of antibiotic resistance on the growth parameters of E. coli. E. coli 0157H7 is a pathogenic strain of E. coli that is known to cause several infection or lead to foodborne outbreaks and illnesses. And then wanted to evaluate the influence of antibiotic resistance on cross-tolerance in this particular strain. I wanted to evaluate the possible mechanisms behind those. And so to do this, we explored some certain assays. So first, we had to develop resistance in E. coli. So we chose two antibiotics, ampicillin and streptomycin, and using two strategies. So one, electrotransformation, where we put a GFP plasmid into the strain. Basically, it's called GFPs are green fluorescent proteins, and what they do is that they when at certain wavelengths, you can see fluorescence. But then this particular plasmid also had a specific loci that conferred resistance to ampicillin. And so we not only had resistance to ampicillin in the bacteria, but we could also see the strains that were resistant through the fluorescence under UV light. And then another strategy was to adapt this bacteria to increasing concentrations of the antibiotics. And by that means, we developed chromosomal resistance. And then, of course, we tested them on different media to see if we had actually developed this resistance. And then we evaluated growth rate. So we took a certain concentration of the cells, about 6 log CFU. We put them in a media TSB, tryptic soy broth, and we grew them at sublethal concentrations of lactic acid. So we had figured out the MIC of lactic acid, and then we took concentrations that were less than the MIC, and we grew the cells in them over 24 hours to see if there are differences in the different strains we had created in stage 1. And then, this was the method we used to determine the MIC. So basically, this is a cell viability method that uses a dye, resazirine. And in the presence of the dye, if the cells are surviving and metabolizing, then you'd have changes in color. So this was a way we could use to determine the concentrations where we didn't see growth anymore of the bacteria. And then we evaluated cross tolerance. So we took all the strains that we had developed and we exposed them to different concentrations of lactic acid at different times. And we evaluated for how much survived. And then we also used an assay, called a live dead assay, which I'm happy to explain later, to evaluate the viability of the cells after exposing them to lactic acid. And of course, we got all the counts and performed the analysis. And then to evaluate what possible mechanism could be causing this tolerance, which we saw, we used a certain dye known as CTCP, which our hypothesis was that the tolerance was on efflux pumps. Basically, efflux pumps are proteins on the surface of cell membranes that prevent the passage of molecules in and out of the cells. So the theory was by blocking those efflux pumps, we wanted to evaluate if the bacteria still had the tolerance that we saw to lactic acid. And so we used CCCP, we blocked those efflux pumps, and we evaluated for survival in 2.5% lactic acid. And this was the results we got for the growth rate. So, just right out of the heart, we could see that there were differences in the growth rate between all the strains. So we looked at things like the lag time duration, so the time the cells spent in the lag phase. We looked at the maximum cell growth rate, and we saw some differences, especially for bacteria that had resistance to ampicillin and streptomycin, and for those that had resistance to ampicillin. And so this was just a graph that outlined those differences, because we compared them and we looked out for significant differences between, lag phase duration, the maximum growth rate, and the maximum population density of the bacterial strains. And these were results that we got for exposure. So we exposed them for 30 seconds and 300 seconds, basically to mimic what we have in food production. So, when you think about the production process, no one really has the time to soak a food product, or in this case, beef, for, let's say, 30 minutes to kill off the bacteria before, they take it out for consumption. Number one, you could have effect on the product quality. But number two is also not economical. And so the industry is always looking for things that are quick, very, that are quick acting. And so we chose these times to kind of mimic what we saw in the industry. And we found that two of the strains, again, the one with resistance to ampicillin and streptomycin, seem to do better when exposed to 2.5% lactic acid and 5% lactic acid. The same thing we saw, the same results, we saw them with the cell viability assay, live dead assay. And so we looked at, the results for the exposure to CCCP, and we saw that when we added CCCP to the media, we kind of abrogated that tolerance to lactic acid. So it was sort of an aha moment. Okay, efflux pumps seem to be contributing to this resistance, but again, that wasn't totally the case for the strain with ampicillin and streptomycin resistance. So there could be something more that was causing the cross tolerance. And so we drew up some conclusion, and some of them were, okay, resistance to ampicillin and streptomycin could lead to some alteration in the growth parameters of E. coli. And, even though the minimum inhibitory concentration did not change, when we looked at things at the sub-minimal inhibitory concentration, there seemed to be some differences between strains that had taken up resistance to the antibiotic versus the parent strain that didn't have the resistance. And, we found that one of the strains, E. coli 0157 H1730, with resistance to ampicillin and streptomycin, was the most tolerant to 2.5 and 5% lactic acid. And, based off of what we saw from the results, it seemed like efflux pumps were playing a big role in the cross tolerance. So we looked at other things because now, we figured out that it seems like efflux pumps seem to be playing a role, but we also found that efflux pumps don't just play a role in cross tolerance, but they could also play a role in something like biofilm formation. If we have a different phenotypic expression for the bacteria, then other things too might be altered. And if you know something about biofilms are a big problem in not just the food industry, but also in healthcare. So biofilms basically are a group of microbes, bound together in this exopolymeric matrix. And it's a way for microbes to kind of protect themselves as a community against harsh environmental conditions. And so our hypothesis was that the bacteria that had acquired resistance to antibiotic might also be better at forming biofilms. And so basically the objective was to determine the influence of the antibiotic resistance on the biofilm formation of E. coli. So we used two classic assays. The first one is called a crystal violet assay. So basically you grow the cells on 96 well plates, they form biofilms. And then using, at this point, this is sort of a novel technique. So basically you use steam to wash the plates, to wash off any cells that are not adhered to the biofilm, layer, something we call planktonic cells. And then we stain with crystal violet and we read off the OD, decolorized and read off the OD at 570 nanometers. And then a different technique we used was confocal laser scanning microscopy, where again, we developed the biofilms. And then we used the confocal microscope to basically take pictures of the biofilms that had formed after we had stained with dyes. That could help us, see the thickness of the biofilms and, different biofilm characteristics. And we found that through crystal violet assay, again, the cell with antibiotic resistance to, ampicillin and streptomycin did better than most of the other strain variants in terms of its ability to form biofilms. But then also from the pictures we had taken, again, the same strain, when we compared the maximum thickness, the bio volume of the biofilms, we saw that the cell with ampicillin and streptomycin resistance seemed to do better or seem to form better biofilms. So the next question was, what about other pathogenic strains of E. coli? So I've been in a different session here where they've spoken about antibiotic resistance and E. coli, and there was a little bit of a conversation about the pathogenicity of E. coli. Yes, some E. coli strains are not pathogenic, and some of them are part of the microflora of the gut. But then there are some strains of E. coli that are pathogenic, especially when you're talking about specifically 0157 H7. That has been the big player to foodborne illnesses for a very long time. But then, recently we've had other strains of E. coli that are pathogenic. And so, wanted to see if we could see the same phenomenon with this other strains. So strains like 026, strains like 0121, we have other strains like 0131. These are all pathogenic strains of E. coli. But then for the sake of this study, we used 0157, we used 026, we used 0121. And we developed the same sequence of antimicrobial resistance in these other strains. And we did essentially the same study. So we evaluated their growth rates and we compared them, we exposed them to lactic acid at different concentrations sublethally, and we looked out for differences between each of the strains. And we expose them to lactic acid, 2.5%, 5% for 30 seconds and 300 seconds, and we evaluated for differences. And we found that basically the strain, our 0157 strain with resistance to ampicillin and streptomycin, again, had better cross tolerance to lactic acid. While we saw some differences between other strains, so 0121 and 026 seeming to do better, that's strains with antibiotic resistance, it wasn't really significant. The main results we found were in this particular strain, 0157H7, H1730, with resistance to ampicillin and streptomycin. And so we came to a general conclusion that 0157H1730 resistance to ampicillin and streptomycin demonstrated higher tolerance to antimicrobials. We didn't necessarily see a significant difference for other serogroups, so we cannot necessarily come to a conclusion that there was increased cross-tolerance in other serogroups. And then as a side project, we saw some filament formation. So as we're undergoing the biofilm formation stage, as we're looking at biofilm formation, we saw that the cells seem to form aggregates. And that was something that was noteworthy. So, cells seem to form better filaments in the presence of these antimicrobials. And it seemed like cells with antibiotic resistance had better filament formation than the cells without. And we also concluded that the higher tolerance that we saw may have resulted from efflux pumps. Again, I'm very sure one of the questions would be, what are efflux pumps and how do efflux pump works? How do they work? So I'll be happy to answer that at the very end. But then we saw that there were some genetic modifications on the efflux pumps, and we saw higher tolerance in cells that had a resistance to ampicillin and streptomycin. So I know this was a lot of technical information, a lot of things to digest, but the big question again was, could antibiotic resistance contribute to sanitizer tolerance in sugar toxin producing E. coli or pathogenic E. coli? And the big answer was yes, the potential does exist. Even though this potential might depend on the bacteria, and even within the bacteria, it might depend on the strain. So, when you look at something like Salmonella, there are over 2,500 serovars of Salmonella. And so it is possible that each serovar can behave differently from the other. So let's say if you're looking at something like Salmonella typhimurium, you might have a different response in terms of antimicrobial cross tolerance than something like Salmonella enteriditis or something like that. In the same vein, when you look at E. coli 0157 specific strain could act differently from, let's say, 0121 or 026 or 0131 or 045. They could all have different mechanisms or different resistance to antimicrobials. Now, a take-home message, which is basically, if there's nothing else from this presentation that you can take home, it's antimicrobials must be used at lethal concentrations, because sublethal exposure to antimicrobials can result in resistance and can result to tolerance. And so the big thing is all antimicrobials, be it sanitizers, be it antibiotics, must be used at the lethal concentration in order to prevent this phenomenon of cross-tolerance or cross-resistance. Thank you. And so at this point, I'll open up for for questions.
Thank you so much, Doctor. That was an amazing presentation. And okay, if you have questions, you can kindly unmute or write it in the chat box and I read it out. You can drop your question now. If you have any question, you can unmute.
Okay.
Drop it in.
Good evening.
Good evening.
Thank you very much.
Thank you, Dr. Ike, for your time. Although I don't know much about antimicrobials, but I really enjoyed the session. So the first question is, was there a particular reason you used 30 seconds and 300 seconds for exposure time? That's number one. Number two, I think you mentioned ammonia and lactic acid as two chemicals that are used as antimicrobial in food. Are there other types? Please mention them. Thank you for the beautiful section. Thank you.
Thank you so much. So yeah, there's a reason we use 30 seconds and 300 seconds because again, in the industry, these are the times that are used. You don't want to really spend so much time because there's cost to spending a lot of times. Things have to move quickly because as they say, time is money. And so when you think about the large scale commercial production, they're trying to get things out into the market and they're trying to do it very quickly. And so most times when you look at those industrial processes, those food products don't spend a lot of time, in the presence of the antimicrobial. In beef production, after all the phases of, after the slaughtering, the evisceration and all of that, they spray with the antimicrobials. And so the spray could just last for 30 seconds. Five minutes is pushing it to the max. And so really you want to have antimicrobials that are very quick acting. So within 30 seconds, you want to see a reduction in the microbial population. Within 5 minutes, you want to see a big reduction, depending on the industry you're talking about. And so that really informed the use of those times, 30 seconds and 5 minutes, because that is what is standard in the industry. And then in terms of antimicrobials, yes, we have a lot of antimicrobials. So I mentioned lactic acid, but we also have peroxyacetic acid. We have quaternary ammonium compounds that are used. Some people use nitrates sometimes, but those are more like preservatives. So some people use nitrates sometimes to combat bacteria. And then other methods that are used are things like, high temperature, like high pressure pasteurization and all of that good stuff. So yeah, the major ones that are used, chlorine-based sanitizers. So, a lot of chlorine, even gas, chlorine dioxide, but more like, chlorine in solution. Peroxyacetic acid, quaternary ammonium compounds, lactic acid. So these are some of the chemicals that are commonly used for decontamination in the food industry. I don't know if I answered your question though.
Yes, you did. Thank you.
Yeah.
Okay, thank you so much, Doctor. If you have questions, you can please drop it in the chat box or you unmute. Muinat, I hope you're satisfied.
Yes, I am. Thank you, Esther.
Thank you. If you have questions, you can drop it in the chat box or you unmute. Is there any question?
Yeah.
Okay, Nancy, you can have the floor. I thought you wanted to ask question.
Hello, we can't really hear you.
Maybe he's having a network issue.
Yeah, I think so. No,
Okay, there's a question in the chat box.
okay.
What's the full meaning of CCC?
So I put it in my presentation, but basically it's the cyanide. So chlorocyanide phenylhydrazone. Basically that's what it is. I don't know if
Okay. Mr. Akile, did you get that?
can you hear?
I don't know if you. I
So
heard you, but I don't know if you got it.
okay, so so it's cyanide chlorophenylhydrazone. Can you see it?
I think he has responded in the chat.
Okay,
Said yes.
okay.
So I
Awesome.
remember that you said you promised to explain efflux pumps.
Yeah. So basically, efflux pumps are proteins that are found on the surface of cell membranes. And what they do is that they transport molecules in and out of the cells. And the molecules can be protons. So the molecules can be waste from the external environment to the interior of cells, or from inside the cells to the exterior. And basically, the efflux pumps can also be used to transport big molecules, like let's say sanitizer molecules. So let's say acetic acid, it would probably go through those efflux pumps into the cell membranes. And so what happens sometimes is you can upregulate the activity of the efflux pump so that they pump more things out of the cells, or you can modify them in such a way that they prevent the entrance of molecules within the cells. So sometimes what happens in the terms of antimicrobial resistance is that those efflux pumps are modified in such a way that they pump out things out of the cells more efficiently. In the case of lactic acid, there is a process by which cells are able to use amino acids, and through modification of the efflux pumps, they pump out proteins they pump out protons from within the cell membranes. Because basically how lactic acid works is it gets into the cell and it begins to deprotonate, and it reduces the pH of the internal environment of the cell. And so by modifying those efflux pumps, what you're doing is to pump out those molecules that can lead to the drop in the pH of the internal environment of the cell. And so some bacteria are able to, use this mechanism to prevent the drop in the pH of the internal environment. So basically, efflux pumps are just those molecules at the cell membrane that help to transport substances in and out of the cells. I don't know if that answers your question.
Perfectly. Thank you.
Yeah.
Okay, thank you so much, Doctor. I don't know if anyone has question again.
Hi, Esther.
You
I
can
think
either drop it in
there are
the
a couple
chat
of
box or
questions.
mute.
There are a couple of questions on the chat on the message section, so you can just have a look.
Okay.
Okay.
It says, how can we develop a novel drug to target both biofilm formation and efflux pump? Will this also lead to multi-drug resistance? That's a very interesting question. I would say there are several. Okay, so the novel drug, it depends on the activity of the drug. So I'll give you an example. Things like ampicillin. Ampicillin can work in two ways. So it can either bind to, the penicillin binding protein on the surface of cells, and that way it can prevent the formation of more cell walls, basically in the cell. So they act on cell wall synthesis. And so resistance, occurs by either some bacteria producing things we call beta-lactamases that can get out and break down ampicillin in the environment, or you can modify the efflux pump in such a way that the ampicillin doesn't recognize those receptors on the surface of the cells anymore, and they can't bind, or they can't modify it. And so a novel drug would have to be one that attacks the cells on multiple fronts. So, it can penetrate the cells, maybe reduce the pH within the cells. It can break out the cell membrane so that, those efflux pumps are broken apart. Maybe it can target nucleic acid synthesis, so that the cells do not form, more progeny cells. So it could basically, the drug would have to have multiple mechanisms of action. I don't know that it's possible. It is possible. Nothing is impossible in science, but I don't know that we're there yet to have something that works on several fronts. Now, one strategy that people use is to use several hurdles. So let's say you can use a combination of high temperature and and chemicals, or you can use a combination of like physical sharing and chemicals, or just a combination of different things, so that they are all acting in different ways. In one of the projects I did, I worked with antimicrobial fatty acids, and basically antimicrobial fatty acids, they bind themselves to the cell membrane of cells, and because the cells do not recognize them, they basically break apart the integrity of the cell membranes. So you can use a combination of, let's say, the antimicrobial fatty acid and something else like peroxyacetic acid, maybe. Or something that can penetrate within the cell, like lactic acid, and cause reduction in the pH. So yeah, we can have that. We can have a novel thing to target several points on the cell. Would it lead to multi-drug resistance? If you use it at the right concentration, I think not. The main issue here is that most of these chemicals are not used at the right concentration, and that's why you have bacteria adapting to it. And so if you go to the lab and you find out that, let's say, as an example, bacteria is not able to survive 20 micrograms of streptomycin, then you'd have to use concentrations above that and nothing less. And so that's why we emphasize use your antibiotics and use your antimicrobials at the right concentration and the right time, so that you don't have development of multi-drug resistance. I don't know if I answered that question, Nasruddin.
Yes,
And
thank you
then,
very much.
yeah, and then how do we curtail the threat? Again, it's all use antibiotics, use antimicrobials responsibly. When I was in Nigeria, my mom, she's a pharmacist, and I would have people come and they would buy these antibiotics, something like ampicillin, and they would just take 1 or 2 tablets and that's it. But that's not the full course. That's not how it's supposed to be taken. What happens is, if you don't complete the full course of the treatment, then bacteria is able to survive that little concentration that you send to it, and it's able to develop genes that can lead to resistance. And so that's how you really have development of resistance across the food chain. And we know that bacteria talk to each other. They can transfer their genes from one to the other. And so basically, the way to curtail it is use antimicrobials responsibly. If you cannot do without it, use it responsibly. If you can do without it, don't use it at all. That would be my suggestion. I hope I answered that, Emmanuel. If you're still here. Okay, any more questions?
Any more question? If you still have question, you can unmute.
Okay, so
Okay.
for Paul,
In the absence of
I think there's one question. There's one important
Doctor, I don't know if you have
question.
any other thing to say.
So there's one question from Paul that says, please, my question is, I would like to look at the possible implications in food manufacturing industries where some antimicrobials are used to clean up machineries. And he's talking about suggesting high levels of antimicrobials. So no, I'm not suggesting high level of antimicrobials. I'm suggesting levels of antimicrobials that have been shown to be the MIC, so the minimum concentration where you have a complete reduction in the microbial population. They might not necessarily be high levels, don't need to be high levels, but they just have to be levels that have been shown practically and experimentally to reduce the population of microbes. In most cases, most of those concentrations don't affect the quality of food. So things like 2.5% lactic acid or 5% lactic acid doesn't affect the quality of, say, beef, but it can lead to complete reduction of microbes. So yes, a lot of people who study antimicrobials also study the effect of the antimicrobials on food quality alongside. And so to answer your question, they don't necessarily affect the quality as long as they are used at the concentrations that have been experimentally determined to be the right ones, and they are used at the exposure time that have been determined to be the right ones. So that's the answer. Okay. I don't know if we have any more questions.
Yeah. Good evening.
Good evening.
May I?
You can speak.
Hello.
Hey, go ahead.
Yes, please.
Sure that, okay. Thank you for the presentation, sir. I have the impression that the old generation antibiotics, like say tetracycline, chloramphenicol, ampicillin, and the rest of them like that,
Yeah.
I have a feeling that they don't have any position anymore in the treatment of microbial infections. I don't know if that is true.
You said, sorry, you said you don't have any what?
I want to confirm if they can still be, if they are still relevant today, as you know.
If they are still
Those old ones, like the ampicillin,
yeah.
and the rest of them,
Yeah.
that probably they don't have any role in the management of bacterial infection anymore, since we know that they have been abused over time, and
Yeah.
they probably will not have any sensitivity anymore. So I don't know if that has been studied or confirmed to be true by any study. That is one.
Okay.
Well, I think let me allow you answer that.
Yeah, so I would say that ampicillin is still considered a first-line antibiotic. So basically, is one that is still commonly used for treatment, not just for human, not just for use in humans, but also for animals. So things like ampicillin are still being used. Things like streptomycin are still being used. Yes, we have new generation antibiotics. So something like colistin is a new one that's coming into the field, and colistin is one that is being used right now. And then some newer generation antibiotics that I can't really remember off the top of my head. But yes, to answer your question, those antibiotics are still being used. They are still relevant. They still work, as long as they are used at the right concentration. You know, um, but there's an increasing trend of antibiotic resistance that we're noticing. And so that's why everyone is raising speaking up about it and saying, hey, we need to be very careful about how we use or misuse most of these antimicrobials, because if we don't look into it, if we don't curtail our use of them, then we could have a lot of microbes, pathogenic microbes that are resistant to them. And so basically they wouldn't have any effect anymore. I mean, things like colistin that was just brought, was considered something that didn't have any resistance to it. Recently, a professor found some genes that are called MCR genes that confer resistance to colistin. And so we even know that, yeah, microbes they have the genes to resist colistin treatment. So yeah, I mean, I know I just gave a long-winded answer to your question, but those antibiotics are still relevant, but then there's an increasing trend towards more of resistance in bacteria towards those antibiotics. And that's why we need to be very careful as a community.
Okay.
Yeah.
Thank you so much. Because I work in the community as a pharmacist, and the trend of use of these drugs, I feel sorry for patients who actually need those drugs, and I don't know how to actually stop that, but we are trying our best, or we are doing our best. We hope that everybody, puts hand in this and try to stop antimicrobial abuse. So my second
Exactly.
question is about, there was this, when I was an undergraduate, I read something about the use of bacteriostatic and bactericidal antibiotics
Okay,
that can
great.
be used together, and then
Yeah.
when you use them together, the mechanism of action is like antagonistic or something. So I don't know if that is confirmed, because I have seen prescriptions where we have bactericidal and bacteriostatic antimicrobials being prescribed together for a patient. And then the last question, which is about antimicrobial now for tuberculosis and the those mycobacteria. I understand that the first line agents, well, they are increasing resistance to those first line agents. That's why we have a multiple resistant and extended resistant diseases. I wanted to ask, the second line and the third line agents are like drugs for normal. They are like normal antibiotics. For instance, levofloxacin is the second line agent for tuberculosis and the rest. So what is that mechanism that makes them relevant in antimicrobial management? I don't know. Is there a way they work to also penetrate the what's the cell wall now for these mycobacteria that makes them also effective against those organisms?
So I'm thinking of the best way to answer your question. Typically, so there are different. So as far as I know, there are different ways these classes of antimicrobials act. So they're either acting on the cell wall, or they're acting on protein synthesis within the cell, or they're acting on nucleic acid synthesis. So these are the three that I know that have been studied so far. And so most of the antibiotic classes fall under those three. For things like penicillin, they act on the cell wall synthesis. And, I spoke about that in my presentation. So, they kind of prevents the synthesis of things we call peptidoglycans on the cell walls. And then some of them, they go in and they affect protein synthesis. So something like streptomycin would fall under that class that basically prevents the ribosomal proteins from forming. And then you have some that act on nucleic acid synthesis. I want to believe, I'm not so sure about this because I haven't done a lot of research on tuberculosis and all of that. I'm not so sure about this, but I want to believe that most of those antibiotic treatments would act on either protein synthesis or nucleic acid synthesis, just to prevent, of course, transfer to other generations. I want to believe that those are how they act. In terms of how the mechanisms of resistance and tolerance. So again, most of these are general. And if you look at a lot of studies that have been done, it's either there's modification of specifically efflux pumps. So there are different classes of efflux pumps and they all work on different things. So the same efflux pump that can work to transport antibiotic into the cell could work to transport, let's say, a different molecule, let's say carbohydrates or some nutrients that the cells need. So so basically, if you have a modification of efflux pump, you can affect a wide array of antimicrobials. And so efflux pump would be one thing to look at, and I'm sure a lot of people are studying that. We did a little bit of that. I would say basic studies on that in the project we did, but a lot of people are doing much more in-depth studies into how efflux pump work and how they affect the range of other antibiotics. And to the first point you were making, yes, the advocacy is people, just speak to people not to use, to make sure that they stick to the regimen of the antimicrobials or the antibiotics they're taking, because they might not know how it might not be affecting them personally, but then they pass out feces into the environment. If you have bacteria with those genes, those bacteria can transfer those genes to other bacteria. Somebody else somewhere can pick it up, an animal can pick it up. It's a very cyclic process. And so the advocacy should be, hey, if this is the stated regimen of taking your antibiotic, make sure you stick to it. I know my mom did this, back, in her practice, everyone had to take the antibiotic to completion and at the right concentration and amount. My presentation was specifically to the food industry, and this is the same thing, that is being preached in the food industry. Hey, we use most of these antimicrobials to sanitize the equipment. We use them to clean fresh produce after we harvest them. We use them to decontaminate meat, make sure they are being used at the right concentration and the right exposure time. This way, we can help to combat development of resistance. And I mean, if you can do without antibiotics or other antimicrobials, by all means, do without it. So that would be my answer. I don't know if I touched on all the points that you mentioned. I couldn't keep track of every single thing.
Thank you, sir. Thank
Yeah.
you. It's fine. Thank you.
Thank you, Dr. Ike, for the wonderful session tonight. Please, we still have more questions from the audience. Hello, can you hear me?
Yeah,
we can
I
hear
hear you.
you.
Yeah, do we still have more questions? So, in the absence of further questions, do we have announcement? Martin, wait. Do we have announcement?
Well, so there's none for now. We just want to thank Dr. Ike for the wonderful presentation. Thank you so much for coming tonight. Really appreciate it. Yeah. Martin, is there any announcement from you? Okay, I'm not sure Martin is here, but that will be everything.
Oh, sorry, Dr. My network is acting.
All right. Okay.
Hello, can you hear me?
Yeah. Yeah, I can hear you now.
Yeah, thank you. Yeah.
All right, thank you, Dr. Ike, for your time. Thank you, everyone, for participating tonight. So that will be all for today. See you on Wednesday.
Thank you.
Bye-bye.
All right.